This is us in the fisheries building. And this is planet Earth. But let's rewind for a second, because it didn't always look like this. There were asteroid collisions, numerous mass extinction events, lots of volcanic eruptions, microbial activity and the oxygenation of the atmosphere, and continental drift and land formation that ultimately led to the planet Earth that we know and love today. These essential events that occurred in the early evolution of the planet gave way to habitable environments for all different types of living things. And beneath the sea surface, there were remarkable changes taking place. Fish that used to look like this in prehistoric times were adapting and morphing and taking on new and sometimes even unpredictable forms. Natural selection, mutations, gene flow, and migration, when combined, played a role as driving mechanisms of fish evolution. Take the humble flatfish, for example, and his other flatfish friends. What makes them different from each other? And what makes this flatfish different from this one or that one, and so on? These are the kinds of questions evolutionary biologists ask themselves. As the tree of life expands and organisms continue to evolve, it is important to understand the phylogenetic relationships amongst different species. The hypothesis of these relationships can be visualized in phylogenetic trees. Building these trees can be based on methods of morphology or genetics. Morphology is basically any qualitative observations of the fish, like fin length, shape, and location, or total body length, eye size, and location. With this information, you can make a hypothesis tree based on shared characteristics. If you want to test this hypothesis, you can take your fish specimens to the lab where you can extract the DNA from the fish using PCR to target specific genes. These target genes could code for proteins involved in essential processes such as transcription and translation or meiosis during oocyte development. Once you have these gene sequences, you can examine how similar or different the genes are from each fish specimen. Are there a lot of mutations in the base pairs, or does the gene seem to be highly conserved? The longer the gene sequence is, the better, because it can allow for more accurate results. The Mega7 software can analyze these sequences and generate phylogenetic trees with bootstrap values as a confidence interval. Bootstrap values that are above 70 show the most likely relationships. The next step is to compare your hypothesis tree to the gene trees and determine which one you think is the most accurate. Based on more data, you might decide to reject your hypothesis tree. The gene tree might show more likely evolutionary relationships between the fish. It is almost impossible for evolutionary biologists to determine if two fish are closely related to each other just based on the way they look. Which is exactly why morphological approaches to phylogeny can't paint the entire picture. When building phylogenetic trees, using shared morphological characteristics in combination with genetic methods can yield the most accurate results.